This lecture should add a level of complexity to what you know already that will surprise some of you as to what, how strange genomic information can be interpreted. And I will tell you that people have looked for some of the mechanisms that I will describe in the brain, because as you well know, new biology is to try and understand how the brain works. I don't know if you realize this, most people in the entire country somehow manage to gear their research to neuroscience in some way or another, because we still have absolutely no understanding whatsoever how memory forms, how the brain really interprets the outside world. We have crude understandings, but we have really no understanding of how long-term memories are formed, short-term memories are formed. And so people have looked at genetics as perhaps one of the keys to it. Perhaps, for example, every cell in your brain is different. So people are studying that in very, very great detail as a way of trying to figure out, is that how memory forms? And the immune system was one of the sort of templates by which people began to study the brain at and see, is genetic modification in the brain one of the ways that the brain makes, I don't know if you're aware, the brain has billions and billions of neurons that help interpret the world, and perhaps many of those billions and billions are different from each other, and thus that's how you might get all those unique memories. So it's a really important system. I will say that, in fact, there is nothing like it in any other cell except for the immune cells and actually some viruses that do some of these tricks as well. So, and some parasites do it. So other than that, we're going to go back to where we started last week, which was the immune system. And I was teaching you just a few, I hope everyone's read through the chapter now, and I hope everyone is training themselves to the word. Antigen, again, was just a molecule that you form a B cell response to, essentially an antibody response to. B cell is the cells that fight infection. T cells help the B cells fight infection. We have what we call an adaptive immunity. And I said this to you guys last week, which is essentially you get an infection, you make a B cell, all the B cells, you can make millions and millions, if not billions of versions of different B cells. And as you make a B cell to fight a particular infection, you're able to improve on that B cell to fight that infection better. So as you may well know, you get a cold, it takes about a week or two to resolve. If you've never had the cold before, it might take you 3 or 4 weeks to get rid of, because it takes about 3 or 4 weeks to build an immune response to something. However, once you've built the immune response to it, you store that information as what's called memory B cells. So just a small set of number of those B cells are stored so that in case you see that disease again, you can mount a very, very quick attack on that disease. And so, but however, every time you get a new disease, for example, even if it's similar to the old one, you mount your B cell attack based on the memory B cells, and you improve on it. And that's what's called an adaptive or acquired immunity. You start making a response, and you are able to improve on that response. Again, what really drives this whole mechanism of antibody response or B cell response is what's called the B cell receptor. And it's the receptor that is initially expressed on the cell surface of the B cell and then eventually secreted in the bloodstream. Again, T cells help coordinate this attack, and they have an equivalent structure called the T cell receptor. And an antibody response is what I told you. There's also something called an innate immunity. Yes?
Can you clarify what
I will clear that up. So there's also something referred to as innate immunity, meaning that if you didn't have your B cells or T cells, you're able to fight off infections because many of the organisms that you're trying to fight, many of the bacteria, many of the things, have particular groups on their cell surface. And there, I believe it's, here's a list of some of the different things you can find. For example, here these are found, this is a microorganism, this is the location of the molecule that is where a particular thing is found, and this is the molecule or the part of the molecule that is you mount an antibody response to. So you're able to not mount antibody response, you mount an innate response to. You have a set of receptors that can already identify those. So this is set, so it gives you sort of a preliminary way to start getting rid of the crap that infects you. However, you may not be aware of this, you cannot tolerate more than 1 or 2 bacterium growing in your body, within your bloodstream. You have more than a couple of bacteria growing, you automatically start getting sepsis, the bacteria secrete stuff, and start killing you. And I don't know if you guys realize this, sepsis is one of the #1 killers, and it's quick. Sepsis kills you in 24 to 48 hours. So if you can't, you probably read about these E. coli outbreaks. These people die. They cannot mount an remember what I said about an antibody response. You got it takes days to weeks. E. coli double every 20 to 30 minutes. You are dead if you can't mount if you can't get rid of a lot of it, and so you get antibiotics to help you out. But sometimes the antibiotics cannot kill the bacterium fast enough, and you die. Yep.
How does the antibody receptor help your immune system boost to kill the bacteria?
We'll get into that in a moment. It's coming. So this is the innate system where you have a set of receptors to help get rid of the bacterium, and it's good, but it's not great. So, and again, you don't have to, I encourage you not to memorize it all, just because where I really want you to understand this stuff is the B cell receptors, your adaptive immunity. So there's one important type of T cell, it's called a helper T cell, and what it does is it produces signals that allow the B cells to differentiate. So many of you know HIV destroys the T cells, and without the T cells you can't mount an antibody response. Even though your B cells are just functioning just fine, your T cells are gone, your T cells need to communicate with the B cells in order for the B cells to fight infection. So this is what sort of your adaptive immunity looks like. You have helper T cells here, and you have B cells. And B cells right now here are attached to the membrane of the B cell. There are trigger, which we'll get to in a little bit, which allows these cell receptors to be secreted. When they're secreted, they bind the antigen. The antigen is dragged to the macrophages, and the macrophages destroy it. And eventually, you end up presenting some of the destroyed peptide back to the T cells in order to make a better B cell receptor. So that's part of the mechanism. But so this is sort of what this is another part of the immune system, the sort of called the cytotoxic T cells. These are responsible for cell media response, which the fragments of the form, this is what I was telling you, the fragments that were generated now get presented back to the T cells, and these T cells now present them back to the B cells. So it's a system by where you can take a foreign substance, destroy it, break it down into small fragments of protein, get it back to the T cells, and present it back to the B cells, and the B cells now make a better B cell receptor. So one of the ways that it makes a better B cell receptor is that the T cells emit a signal, which if you have a good match with a B cell receptor, the T cells secrete a signal that make the B cells grow more, double more. So you now create more and more B cells of that exact same type. And that's called clonal selection. That's the idea that the T cells support the growth of certain types of B cells. And that's basically shown here. Here you have an antigen bound, and that this particular one which binds to this particular B cell now is amplified. This amplification actually is T cell dependent as well. So what is it about the B cell and T cell that makes this all work? It's the B cell and T cell receptors. They are expressed in a very interesting fashion. So I'm going to show you this picture, and I'm going to give you a small video after I show you this picture. So this is what a B cell receptor looks like. B cell receptor, or what we also refer to as the antibody, is composed of 2 light chains and 2 heavy chains. And these are, the light chain is cross-linked to the heavy chain here. Heavy chains are cross-linked to each other. And again, the light chains are cross-linked to each other. These are where the antigens bind to the B cell receptor. It's between the heavy chain and the light chain within this domain here that's shaded red. And these are what we call our antibodies. So the B cell receptor will eventually become our secreted antibody. And I'm going to show you a very brief video on this.
Antibodies of the immunoglobulin G class are Y-shaped glycoproteins that circulate in the bloodstream. They bind to antibodies of the immunoglobulin.
It's loud. I'm going to drop it.
On the lower right corner. The lower right hand corner. Yes.
Immunoglobulin G class are Y-shaped glycoproteins that circulate in the bloodstream. They bind to and inactivate foreign molecules, the antigens, and mark them for destruction. Each IgG molecule consists of 2 light chains and 2 heavy chains. The heavy chains have carbohydrates attached. The regions of the antibody that bind to antigens are located at the very tips of the 2 arms. Each arm of the antibody is composed of 4 domains. 2 are called the variable domains, contributed by the heavy and light chains, and hence called VH and VL. The variable domains are attached to 2 constant domains, again 1 each from the heavy and light chains, and hence called CH and CL. Variable and constant domains share a similar structure called the IG fold. Each domain consists of a pair of beta sheets, one with 3 strands and one with 5. A single covalent disulfide bridge holds the 2 sheets together, which results in a rigid and very stable domain. As their name implies, the variable domains vary in amino acid sequence from one antibody molecule to another, thus providing the vast diversity in structure required by the immune system. The antigen binding site in the variable domains is composed of hypervariable loops that are especially susceptible to sequence variation. The sequence variations in the hypervariable loops are responsible for the specificity of antibodies to particular antigens. Antigens bind to the tip of each antibody arm, generally 2 molecules per antibody. In the example shown here, The antigen binds to the antibody via a large contact surface, providing a tight and highly specific association.
So this is the driver that allows you to recognize antigen. Is this set of so-called light chains and heavy chains. These are made by different genes. So we're going to look at the light chain. So in humans, which is probably all you care about, is there are two types of light chain genes, the B kappa and the B lambda. Those are two different gene clusters, B kappa and B lambda. For the heavy chain, there's only one locus. This is an example of the B lambda chain gene. The B lambda chain gene, for example, is constructed in this manner. And again, you're going to hear a very simplistic version, and then it's going to get more complex in a moment. So here you have germline, you have what's called a leader sequence, an intron, a variable domain. Here this is a J segment. J stands for joining. You have a variable, a joining, and a constant segment. And as you see, this is the germline configuration with introns. Notice there's an intron in the leader, between the leader and the variable region. And this whole region here is called V lambda. So V lambda has a leader sequence, an intron, and then the variable portion of the gene, followed by more introns, plus the J segment, more introns, plus the C. One of the things that happens, which we'll go into great detail coming up, is that the genome between the variable region and the J region is deleted. This is genomic deletion. So unlike what we talked about introns, per se, being deleted in the RNA, this region here is deleted in the genome, gone. And that's called DNA rearrangement. Once that occurs, you've now fused V to J, sometimes in frame, sometimes not in frame. As you'll see very shortly, where it joins, how it joins will decide whether or not V and J form a proper protein or proper open reading frame or not. Once V and J have joined, then you make an RNA, that RNA gets spliced, and then you make your protein. However, one of the things you should note, which is extremely important here, is that the VJ joining need not make a productive protein. That joining is sloppy. So you can have it in frame, you can have different amino acids at that point of junction, and it's sloppy. This doesn't sound like a lot of variability to it because, yes, you have one point of sloppiness here, but it's not quite enough to make the antibody diversity we have. And what you need to know is that, in fact, you don't just have one V lambda region, you have about 10 to 20 of them. You have a bunch of J regions as well. So in fact, it's not just one V joining to one J. You have a whole bunch of different V regions that each have the possibility of joining with a different J. And all those joinings are by DNA rearrangement. You lose genomic DNA. This means that B cells and T cells when T cell rearrangement underoccurs, but B cells do not carry all the genetic material that the rest of your cells do. It is the only cell in your body B cells and T cells, or immune cells, that do not carry the entire genetic information of like all the other cells in your body. Yes?
Now, is this method of DNA rearrangement the reason that B cells have such high specificity for the antigen? Now, because they're not able to change their receptors later on in the future, so this is the reason why, because they've deleted portions of their genome?
So the question is the deletion the mechanism by which specificity occurs, that they're not allowed to change it over time? I don't know. I don't know. The reason why I say that is there are other ways to drive this diversity that can change over time. So I can't swear, I would not know how to say yes or no to that answer. So, as I said, and what's important you understand is, remember these are B cell receptors. How do B cells get out of the cell? They have to be targeted to the membrane. That's why there's this leader sequence. That leader sequence is part of the protein. That leader gets spliced on to the variable region. However, when you finally get to the mature form of the protein that's on the cell surface, that leader sequence has been cut off. Because that was only used to target it to the membrane. So you have this very complex structure of a leader that splices onto variable, that you have genomic rearrangements between multiple V regions and multiple J regions, and you have two different loci for this for variable regions, a lambda region and a kappa region. The H chains, the heavy chains, are even more complex. In fact, the H chains are so horrifically complex. This is just an introductory view to the heavy chains. The heavy chain genes, for which there's only one locus in the genome, again, there are an array of these V regions. There's an array of D regions. There's an array of J regions. And actually, there are several constant domains as well. So this is just one version where you have a leader, because you've got to get it out of the cell membrane. Intron, V domain, the variable domain, the B and the D will eventually come together by DNA rearrangement. However, the very first event in heavy chain rearrangement is deletion of D, one of the D segments to one of the J segments. So you have what's called DJ joining. This is a stepwise process where first D's and J's join by rearrangement. Just as it says here, DJ. Once DJ is formed, and it doesn't matter if it's productive or not, remember what I said is that every time you have one of these DNA rearrangement events, you may take a protein and put it next, part of an open reading frame next to another open reading frame and make a non-functional protein. So for every one of these DNA rearrangements, first the DJ joining, then ultimately the VD joining, you can make unproductive proteins. There's a lot of unproductive proteins to make by DNA rearrangement. Finally, after you have the VDJ joining, you then undergo splicing, and even the C region has splicing that it deals with. So this is what the lambda cluster looks like. The V lambda has a whole bunch of V genes, has some J genes here. And notice there's J lambda 1, J lambda 1 constant region. They come as pairs. But here in the kappa region, you have a whole bunch of V's, another bunch of V's, a whole bunch of J's, and one constant region. one of the things that you'll be surprised to know is that you only make every functional B cell makes one functional light chain and one functional heavy chain. And I'll try to describe this to you as follows. So, you have in your cell V kappa, the maternal and paternal allele, V lambda, these are the light chains and the heavy chains, loci, the two light chain loci. And you have V heavy chain and V. I'm just using the V to abbreviate the whole cluster. Okay? So okay, so you have the two kappa clusters on each maternal paternal chromosome, the two lambdas, and the two heavy chain clusters that are available to you. As I said, the first step is you do DJ joining on the VH, on a VH region for one of the two for both of the chromosomes, for both chromosomes. Right, so here, for example, let's draw it out in its glory. You have a set of V chain, V's, you have V's, you have J's. One of these D's joins to one of these J's, and that's on both chromosomes. So you have this event going biallelically. And it's random. So maybe this one on this one. Okay? So you have DJ joining as the first step in the heavy chain loci. This is a deletion of genomic DNA. You're deleting out everything in the middle here. You're deleting out everything in the middle here. This is deletion of genomic DNA. It's gone from the cell. Once you've done this, yes? I'm just ignoring it for now. The constant is down here. First step is DJ rearrangement. You do not know from either chromosome whether this DJ rearrangement has created an in-frame open reading frame, right? So there's some nucleotides here, some nucleotides here, and you do not know whether these two have spliced in a way that haven't incorporated a stop codon. These are genomic rearrangements. Maybe a stop codon got in there in the same reading frame, and thus it's a dead protein. You do not know. The next step, step 2, one of these chromosomes undergoes V to D rearrangement. That means all this is deleted. So VD VDJ, I should say, rearrangement is step 2. And that's on one chromosome. Now, the system tests to see if it's made a functional protein. It does so because when V, D, and J come together, and then you get VDJ, C, you have your leader with the intron here, you have introns. The C region has introns in it. C, A, B, C, right? These are just introns. You splice it all together, you make the protein, you splice mRNA, you make protein, Does it go to the cell surface? If the answer is no, it does not go to the cell surface, the cell goes back to the drawing board. And it will now rearrange any of the V's with any of the D's. And you will get a VDJ rearrangement again. And you'll test again. And if that fails to form a B cell, a secreted receptor, a secreted protein, or a membrane-bound protein, I should say, the cell dies. Bye-bye. All done. You have two chances at getting it right. If you fail, the cell is done, and you are trying it again in another cell. Yeah, there's a question back there?
I know you answered it.
There was another question back there.
So when it goes back to the drawing board, now there are fewer D's and fewer J's to work with?
No, it's the other chromosome it works on. That chromosome is done. Now it works on the other chromosome. This is called, so you're testing the ability of an allele to function. Once you have an established VDJC protein, now one of the light chain loci begins to rearrange. And the same thing has to happen. You make a V to J, you test it. If it works, great. If it doesn't work, you move on to the next allele. If that one doesn't work, you have a chance to go to the lambdas. If none of them work, you're done. Bye-bye. Bye-bye, cell. You have to get one of those events to occur. So you can see that you are making isotypic receptors from cells. One type of receptor can be made. You're not allowed to, if, for example, the first one were functional here, you do not get rearrangement on the second chromosome. You make one type, and that's because the machinery that's used to make this rearrangement is gone from the cell. Once the rearrangement has been made, those proteins that cut the DNA are gone. Yep.
Does that chromosome get methylated so that it can be expressed?
Actually, it is expressed, but it never makes a functional protein because it's the nuance. You can't get DJ recombination without expression. It's a nuance of the system. Don't need to know that. So let me take a minute, and I'm going to show you another movie.
Recombination. Immunoglobulin genes are composed of separated segments of DNA that become joined together by immunoglobulin genes are composed of separated segments of DNA that become joined together by a process called somatic recombination to make a functional gene. In heavy chain genes, there are 3 gene segments: the variable or V segment, a diversity or D gene segment, and the joining or J segment. Light chain genes, such as those shown here, have only 2 gene segments, the V and the J segments. Gene segments that can be...
Notice I want to show you something here in the picture.
Gene segments that can be of only 2 gene segments.
Notice what they're showing you here in this picture. They're not showing you, but there's a leader here with an intron. This is the leader portion of the protein with an intron in between it for each of the V segments. Each of them have their own leader sequence as a separate intron.
The V and the J segments. Gene segments that can be recombined have specific sequence motifs adjacent to them, called recombination signal sequence, or RSS motifs. A protein complex containing the products of the recombination activator genes, RAG1 and RAG2, binds specifically to the RSS motifs. In this example, flanking a V gene segment and a J gene segment. The individual gene segments, to whose flanking RSS motifs the RAG protein complexes bind, are selected at random from a number of copies present at each gene locus. The rad protein complexes bring together the gene segments to be recombined and cleave the DNA exactly at the junction of the gene segment and its adjoining RSS motif. The cleavage creates a hairpin of DNA at the end of the gene segments and double-stranded breaks at the ends of the RSS motifs.
So you see what it said, is that normally this is double-stranded, but the cleavage mechanism actually binds this top strand to the bottom strand, and same here. So these are actually joined here, whereas these are unjoined at this point.
Additional proteins, DNA-dependent protein kinase, COO, Artemis, and a dimer of DNA ligase and XRCC4 are incorporated into a large complex with the rad proteins. These RSS ends are joined, forming what is called the signal joint, to create a closed circle of DNA that plays no further role in the recombination process.
It just gets lost, gone in the cell.
The DNA hairpins at the ends of the gene segments are then cleaved. An additional enzyme, terminal deoxynucleotidal transferase, or TDT, is recruited and adds additional nucleotides to the ends of the DNA strands. The other enzymes in the complex ligate together the two ends of the gene segments, completing the recombination process.
Notice when TDT comes in, where it normally cleaved it right in the spot, the rejoining mechanism adds a set of random nucleotides in the middle. So the mechanism of joining creates randomness. And that's why I said sometimes they're in frame, sometimes they're out of frame. You can't predict how many nucleotides are added or which nucleotides are added. I think that's the end of the slide. So are there so far any questions about what I just showed you about DNA recombination?
Immunoglobular.
Like for the heavy chain, how many exons are there that end up on the mRNA?
A lot. I don't remember because the heavy chain is broken up into many exons.
What about
And the light chain and the V part has at least 2 exons, the leader portion and the variable portion.
So does the joining together actually create one exon together, or is
No, the joining only deletes the region between the D or the J or the V or the J. And the
So is that randomness over there? Is that still part of the exon, or that?
yes, it becomes part of the exon. Yes. So this has become, yes, that randomness becomes part of the exon. I'll get to you in a second. Yeah.
Yeah, my question's kind of similar. I'm a little confused. Each of these segments, the B, the J, and the E, are they considered exons of a gene, or they're?
Yes.
Okay.
Each of the V, J segments are considered exons of the gene. That's correct.
And then, but is there a way to refer? Is there a name for the V or J or V segment? Or do you also?
They call it the immunoglobulin locus.
Okay.
Either the heavy chain locus or light chain locus. Yep.
Clarify, the random nucleotides that get added in are just the ends when the fusion actually happens?
Yes.
So wouldn't the enzymes that bind and cleave bind to specific sequences, so why is it so random? Like wouldn't it be doing the same?
So if you see here from the image here, let's go back to that image where it cleaves. So you see this is the cleavage event. So here, this is a perfect cleavage event right where the RSS site was. However, when you open up the bridge between the top and bottom strand, when the TDT comes in, it adds random numbers of nucleotides. It adds nucleotides.
Pins at the ends of the gene segment.
Right, so it adds these. So it adds these random nucleotides and then rejoins. There's another question, yeah?
The original DNA binding protein that recognizes the motifs, I'm guessing the motifs are flanking all these.
Yes, I will go into that a little bit more. Yep. You?
What prevents this system from just recombining constantly
Yeah, I actually don't remember all the details, but, so that's what's heavily studied is why is it that you can recombine and then test? So one of the things that I didn't tell you is that how is it you're able to test is actually quite an important thing. So here, let's go back to, for example, the heavy chain. You have V's, you have D's, you have J's, and you have C. You actually have a very strong enhancer back here called the mu heavy chain enhancer. That's all the way back in the constant region. And so as I said, initially, D's and J's are joined. And then the trigger is that there's a trigger where one of the two alleles, where one of them joins the V to D, somewhere, one of the V's to D's. When you delete all this DNA, you bring the heavy chain enhancer close to the beginning of the V, and there's a TSS associated with every V segment. And thus you test. And the driving of this transcription seems to be what limits the ability for the other allele to be rearranged. So it's this enhancer then allows this to be functional. It wasn't allowed to be functional until one of the V's rearranged with one of the D's. Yep.
How is it determined which allele is going to be expressed first?
It is not, it's random.
So both of them could be expressed at the same time?
No, because it only could be expressed at the same time if one of the alleles is non-functional. Remember, you do not rearrange the other allele unless you make a functional B cell receptor. It means it gets out to the cell surface. Right? You're recombining V to D to J, and you have a splice leader here as well, and you have introns in your constant regions. Now you splice this all out into a functional mRNA, and now the question is, do you make a protein? That protein has to get to the cell membrane. If it fails to get there, it rearranges the second allele. If it gets there, the second allele is not rearranged. There's another question somewhere here? Yeah?
I was just going to ask about the mechanism, how to understand the protein is functional. Because is it just how it interacts with the membrane and you put on the membrane? Because that's not saying anything about the receptor, it's just saying how can you put it on the membrane. So like the receptor itself might not be able to find anything. It's still functional in that it can be
So there are a couple schools of thought. There's some schools of thought that there's signaling from the receptor back into the cell saying, I work. Right, that it couples to some signal transduction cascade which shuts down the system. There are other schools of thoughts as well, and there are lots of debates about how this works. But one school of thought is, a simple school of thought is, the receptor gets to the cell surface, is able then to send a signal back to the nucleus saying, success. That's the most simplistic version. And I'm sure Laurel Eckhart would have a more complicated version of that in her class. So now let's go through this a little bit more. So this is what recombination drives this diversity. And now you're going to see a lot more detail about how this works. So again, now this is what it looks like in all its glory. The heavy chain locus, there are 300 VH regions. There are about 20 D regions. There are about a bunch of J regions. And notice here, these are the constant regions here. The mu, the gamma, the mu, the delta, gamma 3, gamma 1. This is pseudogene, alpha 1. Another pseudogene, gamma 2, gamma 4, epsilon, alpha 2. I don't know if you remember in Bio203 or something, you've probably heard of IgG, IgE, IgD, IgA. These are the different types you can get. So once you have your BDJ working, you first make the mu form. The mu form is the membrane-bound form. That is the one that you test. The secreted forms are all the others. So you have to first splice, you have to first do the splicing reaction with the mu form. That's the first step in producing an antibody, a B-cell receptor. Once you've achieved success, you begin to do what's called class switching, where you actually, it's another DNA rearrangement event where you delete out things between the mu region and an A, or mu and a G, a mu and a delta, et cetera, and you do what's called class switching. There's a question over here.
Yes, basically, so it's the mu form that determines if your MHC type 2 is going to show up on the B cell, right?
The mu form does several, so mu form is the non-secreted form. So it's the one that basically is able to receive information from the T cells. So, but also it has the most important role of saying that it's a functional receptor. So this is the heavy chain locus, and here's just some descriptors of these RSS sequences. So the recombination signal sequence, you're not required to memorize these whatsoever. They're called heptamer and nonamer regions. So in the V kappa region, if you notice, in the kappa chain locus, the heptamer nonamer sequences, here in blue and red, are spaced by 12 base pair spacers on one side, and here on 23 base spacers on another side. So this is the signal. These are conserved sequences, the heptamer nonamer sequences, which constitute the RSS sequence. So the RSS sequence is this whole sequence. It's one part of that sequence has a 12 base spacer in it of any nucleotide. The other end of the sequence has a 23 base spacer on this side. There is nothing really important about knowing that it's 23 versus 12 because it can be reversed and on different genes in different segments. But what is important is you have to join a 12 base spacer to a 23 base spacer RSS in order to get DNA rearrangement. And here you can see, for example, in this particular case, you have these spread out in different fashions between the heavy lambda and the two light chains. Let's see what so here's what I showed you in the movie. The two enzymes that do this effect are called RAG1 and RAG2. RAG1 and RAG2 serve to cleave at precisely between the B's and J's, or D's and J's, whatever, and cause reclosure and reclosure of the DJ joint, plus some random nucleotides in the middle. That's what I showed you in the movie. This is the allelic exclusion is the mechanism I already talked to you about, which is the process where you test. So I really dread teaching you about allelic exclusion, at least the wording, because allelic exclusion implies that the allele somehow knows that it's excluding the other allele. So for example, when I talk to you about here that you make one functional, you don't allow the other allele to be made. That concept is called allelic exclusion. That one allele excludes the other allele from being expressed. The nuance of why that's probably poor descriptor is because the other thing I told you was that allelic exclusion is a process by which you make a protein, you get it to the cell surface, and you signal. allelic exclusion to some degree implies you did something to the other allele, which is wrong. Allelic exclusion is just a mechanism by which one functional allele is made. Because you do not have to necessarily signal to tell the other allele do not recombine. You can just tell the cell, get rid of your RAG1, RAG2, and you won't recombine. So people have misinterpreted the word allelic exclusion. I'm sure your book has as well. Allelic exclusion is classically explained that you're shutting down the expression of the other allele rather than simply not allowing the cell the opportunity to rearrange the other allele.
Do we know anything about the mechanism of how RAG1 and RAG2 choose one?
It is completely unknown how RAG1 and RAG2 choose one allele versus the other. Yeah?
When it forms the circular DNA, when it splices out, it would seem like it's doing that to preserve that piece.
It doesn't. It gets rid of it. It's gone. So the question is, what happens to the circular piece of DNA that's been deleted out? It's gone. So what's really important here that you really have to get through is that you have to incorporate the idea of, I won't finish any more slides today, you have to really incorporate this idea that here, not only do you have splicing of exons together, but you also have DNA rearrangement. This is not mRNA splicing. This is DNA rearrangement. Deleting double-stranded DNA and making the cells unique. Yep. Yeah?
Does that mean that the first time it works, when the first allele, does the second allele remain untouched?
That's correct.
So you have two different alleles, two alleles with different sequences. They're no longer copied.
That's correct. I think you're correct, if I heard you correctly. Once you get a rearrangement, two chromosomes are different from each other.
Right, so that's the amount of
You'll only ever make proteins from one allele.